as Jason said, this talk is going to be on the use of remote sensing, GIS, and spatial modeling for investigating the epidemiology of vector-borne diseases. This is really just one of my research areas. I'm not an epidemiologist, I'm not a virologist, but I work with a lot of them. And so I've developed this talk sort of for a general audience. So if you want to get into any of the more nitty-gritty later on, please do ask away at the end of the talk. So I've divided this talk into 5 major sections: vector-borne diseases, landscape epidemiology, GIS&T applications, challenges, and future directions. And I'll be focusing on the first 3 areas during the talk, but I'll be concluding with the challenges and future directions. So vector-borne diseases are diseases in hosts caused by infectious pathogens and transmitted by vectors. Now, the hosts in this case includes human beings like us and other animals. The pathogens can be viruses, parasites, and bacteria. And the vectors are primarily mosquitoes, fleas, and ticks, but there are also other vectors like snails, for example. The table here shows a partial list of some of the diseases that are transmitted by different vectors. So for instance, the Aedes mosquito transmits diseases like chikungunya, dengue, Rift Valley fever, yellow fever, and Zika, all of which happen to be viruses. But to just give another example, fleas, for instance, transmit the plague, which is caused by bacteria. Now, in this talk, I'll be focusing mostly on mosquitoes because that's where most of my work has been in this general area. In fact, specifically the Aedes mosquitoes and some of these viruses and diseases that you see listed here. Now, why should we care about those diseases? I mean, okay, we are in COVID times, so we do care about disease, I suppose, but why should we care about those vector-borne diseases? Well, there are 12 major diseases caused by vectors globally, and those cause over 700 deaths per year. But that doesn't account for all the people who fall ill from some of these viruses. And some of them are very, very ill. Some of the symptoms include fever, headaches, muscle and joint pains, rashes. And then some of the more severe effects are things like microcephaly, which is a birth defect caused by Zika virus. Now, about 80% of the world's population is at risk of catching one of those diseases. And so I would argue it's an important topic of investigation. Now, what can we do about it? Well, vaccines are potentially one option, and there are vaccines for some of those viruses. But to just give one example, chikungunya, for instance, there's no vaccine for it. And so the next best thing we can do is to try to prevent, control, and manage the vectors that transmit those diseases in the first place. And if you look at various organizations from the more local to the global scale, let's say New Mexico Department of Health to the World Health Organization, they've all developed vector control plans and are actively trying to manage and control those situations. Now, one other question is, well, how do we implement vector control and management in some kind of an effective way? We don't want to, let's say, spray pesticides or herbicides or things along those lines all over the place. So what we really want to do is we want to be able to understand where the risk is highest and when the risk is highest. So you can see here a map from a study done by Michaela et al. In 2018 that shows the risk of yellow fever virus spillover in Brazil in 2008 at 3 different points in time, January, June, and September. And so what you can see, of course, first of all, is that the risk of spillover varies across space. So there's higher risk in the northwestern portion of the country than, let's say, in the southeastern portion. But you can also see that that risk varies throughout time. So there's certainly certain times of year when you're more likely to get infected with yellow fever virus than at other times. So, and I didn't mean to animate it, so I'll just bring the whole thing up. I want to take one more step back. So a lot of those viruses originate in what we call the Sylvatic cycles. So that could be like the forest cycle, the natural cycle. So we have, viruses that may emerge. They get transmitted, let's say, by mosquitoes, but the hosts are non-human primates and other types of animals. What can happen is that a person, for instance, goes into the forest to grab some wood or, get other kinds of supplies, and they get bit by a mosquito that is infected, let's say, with one of those viruses. Well, that person then goes back into maybe an urban environment where maybe another type of mosquito that can potentially also transmit that same disease bites that person and then bites another person. Before you know it, you go from one infected person to many, many infected people. And so there's a possibility that essentially the cycle gets established in an urban environment. When that happens, and it's happened, for instance, with yellow fever virus, we call that a spillover event. Now, the reverse can also happen. So, for instance, the Zika virus originated in places other than South America, but it was brought to South America where it established in an urban cycle. Well, if an infected person with that virus goes into the forest and then gets bit by a mosquito there, and then that mosquito bites, let's say, a non-human primate or some other kind of animal, there's a possibility that that virus then suddenly gets established in the Sylvatic cycle. That's a process called spillback. And that's really, in some ways, you may think, oh, well, why should we care about this? I mean, we should be more concerned about spillover, right? We don't want the virus from the Sylvatic cycle in the urban environment. But the reverse is a huge problem as well, because if once the Zika virus, for instance, becomes established in the Sylvatic cycle, it's going to be really, really hard to eradicate. And so in some ways, when we're talking about trying to predict risk, like where and when it's the highest, we kind of want to be able to predict those spillover and spillback events as well. And that is where landscape epidemiology comes in. So landscape epidemiology describes how the temporal dynamics of host, vector, and pathogen populations interact spatially within a permissive environment to enable transmission. So there's a lot of stuff going on to really understand where and when transmission risk is highest. We need to know something about the hosts, the vectors, and the pathogens. We need to know where they can do well. And so it turns out that it's like this tiny little area of intersection here, which is called the nidus. So the nidus is essentially a place or a spatially defined focus in which transmission happens. Of course, this so the nidus essentially then has certain characteristics habitat characteristics, let's say, that work for the hosts, the pathogens, and the vectors. And so that includes things like climate, temperature, precipitation. That can include topography, geology, soils, vegetation, land use, housing quality. Just to give one example with respect to mosquitoes, for instance, they do have certain temperature requirements, so they can't do well when it's too cold or when it's too hot. They definitely need moisture. But, they do really well, and like an urban mosquito does really well when there are no screens in the window, when there is, some pots maybe sitting outside with water where they can breed, that kind of thing. And so in some ways, I don't know how many of you are geographers here, but to me, this just screams geography, right? I mean, we are looking at interactions between the atmosphere, the hydrosphere, the biosphere, the lithosphere, and of course, people. And so, it's a pretty complex endeavor. And of course, it's not static, right? This nidus, this place where transmission can occur, it varies both over time and over space. So in terms of temporal changes, we see them seasonally, annually, but also over decades. Here's just one example from a study that we did in Senegal. And the details really aren't important, but what you see here is temperature, which does not change much throughout the year. Precipitation, though, changes quite a bit. And you can see here the abundance of different mosquito vectors over time. So there are certainly, certain times of the year when the abundance is higher than at other times. And of course, climate change is really messing with a lot of those things as well. So trying to understand how climate change influences vector dynamics is a really critical topic of research. And then, of course, we also have spatial variations. And so those are in part certainly caused by climate changes, but also importantly, by anthropogenic changes. So here's an example of a land cover map that I just made for the Las Cruces area. I mean, this area, at some point, we had the Rio Grande, we had the floodplain, and then mostly rangeland. what you see here is that this landscape has really become increasingly fragmented, especially by different types of urban land uses. So you can see those indicated here in the red colors. So the agricultural lands, which of course were initially carved into the rangelands, are now being increasingly fragmented by urban land uses. And so that of course affects the spread of disease or the persistence of disease. So understanding those local spatial dynamics is really important. At the same time, oftentimes we're trying to make predictions at the global scale as well. And of course, globally, we are oftentimes now talking about enthromes instead of biomes, because a lot of the world's biomes have become so transformed by human action. And this is just one example of how space and time components can potentially interact. So for instance, urbanization, of course, has transformed landscapes, and we oftentimes have urban gradients where we have highly urbanized areas with a lot of impervious surface. Then we may have some residential areas where we kind of see a mix of houses and roads and driveways, but also vegetation. And eventually we get into the more rural areas. So not only do we have different habitats that have different sizes and shapes and levels of connectivity, but they also produce different types of biophysical feedbacks. And so we actually see differences in microclimate across these urban gradients. And so they influence mosquito dynamics and the dynamics of other vectors that transmit diseases. And so trying to understand these complex dynamics is a huge challenge. And that is why really to address these issues, we need interdisciplinary endeavors. I mean, as geographers, we can do a lot of things, but I mean, like I said, I'm not a virologist. I'm not a mosquito ecologist. I'm not familiar with the literature on all, on that topic. But, then again, the biologists and virologists that I work with, they don't know how to do GIS or how to analyze a satellite image. And so what we do is we come together to try to address these bigger problems. And so one of the projects that we just recently had funded through the NIAID, so I was actually in a Zoom meeting with Dr. Fauci, which was very exciting. But it's this project called Create NEO, which is short for Coordinating Research on Emerging Herboviral Threats Encompassing the Neotropics. And so it's a collaboration between the United States, Panama, and Brazil. And it involves a lot of different people. You can see here an organizational chart. The details are obviously not important, but you can see here in different colors some of the different countries that are involved, some of the different universities within the US, and each box represents one essentially key collaborator, PI or co-PI. So there are a lot of us involved trying to develop a surveillance network that is then coupled with modeling approaches to better anticipate and counter emerging herboviruses. All right, so that kind of gets us a little bit more into the GIS&T thing. So what do we need to sort of predict when and where those diseases might emerge? Well, we typically need species data, and that can include data on the hosts, the vectors, the pathogens. Most of the times it's easier to collect the vectors and then we have to basically see if they're actually infected. But we basically need species level data, and those usually are collected in point locations. Now, there are some databases that exist online. In fact, it's sort of a requirement of most brands these days that when you collect data, you make them publicly available for transparency and accountability purposes. So we can certainly go to these databases and get some data there. There are a lot of published papers that are based on data collected by the researchers, but most of the time we also have our own project needs, and so we basically have to go out and collect our own data. And here are just a few photos from some of the field work I've been involved with. These top two images actually from some field work in New Mexico, where we set different types of mosquito traps in various environments. We'd usually set them out one day and then pick them up the next day. And then here is somewhat more exciting work in Sarawak, Borneo. This is actually what's called a backpack aspirator. So this young woman, Anita, she's got a backpack and this hose, and she's actually vacuuming mosquitoes from the surrounding landscape. And then this young man, Nico here, he has a slingshot to try to get essentially a rope over a tree at a high elevation to set an ovi trap where we're collecting basically eggs from mosquitoes. And so those are the kinds of point data that we need to ultimately try to make predictions across space, because we obviously can't collect mosquitoes or other types of vectors in every single location under a surface. So we have to collect them at points. The other thing that we need, of course, are environmental data that would allow us to make predictions. And so a lot of those data are already available. So the CHALSA database, for instance, provides climate data at the global scale at a pretty coarse spatial resolution, like 1 by 1 kilometer, but it gives you information about average annual temperatures and precipitation in each month and isothermality. There's I don't remember how many variables, but there's a lot of variables that you can just download for free. The Shuttle Radar Topography mission provides global elevation data. And of course, those of you who are familiar with GIS, you know that you can use those data in a GIS environment and derive other types of topographic variables like slope and aspect and ruggedness and curvature and whatnot. The MODIS sensor and NASA provides information, for instance, on vegetation greenness and other types of parameters. So again, that's freely available data. There are also some data on urbanization and human settlements, for instance, provided through CDAC. And then, of course, just to highlight a local feature here, ArcGIS up at UNM provides a lot of data that could potentially be used for more landscape level type models. But oftentimes, of course, we have to generate our own data, and that's where remote sensing comes in, which I'll talk about a little bit here in a moment. All right, so what are some of the GIS&T tools? And by GIS&T, I mean Geographic Information Science and Technology. What are some of the tools that we can use to support this research, to generate the data, analyze the data, and model with the data? That includes the Global Navigation Satellite System, Geographic Information System, or GIS, remote sensing, and spatial analysis and modeling. And there are certainly different ways to organize this, but this is just kind of how I set it up. So how can these tools be used? Well, at the most fundamental level, we can use, a handheld GPS or fancier GPS units to record species locations. And you can see Katie here. She's a former PhD student. She's got her GPS dangling down there. She's collecting some other data. But we need geographic information for the locations where we collect species data so that we can later on tie that to other types of spatial information. Then, of course, data capture, storage management. That's, again, a pretty fundamental thing. So we want both the spatial data, and that includes, again, when we try to model things and predict things, I mean, that's sort of the big thing. But the first step is really to get the data in the first place. And so the species data are one thing. Those are usually collected in point locations. And then to make predictions across space, we need, of course, spatial data across space. And that can include a lot of different types of variables, like land cover, like climate, and so forth. But typically, we bring them all together in sort of a database, and that has a spatial component and, of course, a more aspatial or tabular component. And then we can use really GIS and remote sensing in conjunction to help with sampling efforts. And that this may not sound like a big deal to you, but that's actually really critical. I mean, if our sampling design is poor for the species locations, all the models are going to be complete crap as well. So coming up with a careful sampling design that helps you address the questions that you want to address is really important. And so I'll just give you a couple of examples. So for instance, in 2016, we had a project funded by the New Mexico Department of Health to better understand the spatial distribution of Aedes mosquitoes in New Mexico. At the time, there were 2 observations, if I remember correctly, of Aedes mosquitoes, and that was it. But for some reason, the CDC had actually generated a map that suggested that the species occur like in the whole southern 2/3 of the state. But really, ultimately, we had no idea. So the first year, what we did is we were basically trying to understand if these mosquitoes, first of all, where in the state they occur, and if they are associated with certain types of land covers. And so we basically sort of took the southern 2/3 and I should take a step back. Of course, we have limited resources. I mean, field sampling takes a huge amount of time. I mean, we are located in Las Cruces. We have to drive to all of those locations. You oftentimes have to then go from your car and hike to the location. Again, you have to set the trap out one day, then you have to collect it the next. And so it takes a huge amount of time and effort. And so basically, we had to come up with a sampling design that would meet our project needs, but that would at the same time also help us, that would also be feasible. And so we limited ourselves to the southern 2/3 of the state, because basically we said, well, they are unlikely to occur in the northern part where it's pretty cold. Of course, we included, Santa Fe, because how can you not include the capital of the state? Then we we had a land cover map, and we said, okay, we want to sample in these 6 key categories: urban or built-up land, agricultural land, rangeland, forest land, wetland, and barren land. And then we said, okay, we can't have these points all over the county, because again, we don't have the time to drive around like that. So we said, okay, so they have to be within, let's say, a 10-kilometer buffer area. And then we also said, well, they also have to be within a certain distance of roads, because there's only so far we can hike with all of that equipment. And so based on these various criteria, we basically defined land cover areas in which we could potentially sample, and then we just dropped the points randomly. So we had the random element, we had sort of a stratification element, and we had this element that would allow us to actually do it logistically. Anyway, so those were the sites where we ended up sampling. And so you can see that, remote sensing was used to generate the land cover data, and then GIS was used to implement those different types of criteria to ultimately come up with those points. Here is another example from our work in Borneo. And I know those points look really clustered, and spatial autocorrelation is always an issue and a concern. So in this particular case, what we were interested in was how species abundance and diversity changes with increasing distance into both forests, forest to the right, and oil palm plantations, to see in part, what impact do these oil palm plantations have on mosquito abundance and diversity. And so basically then we digitized oil palm plantations and forest areas in the study area here in southwestern Sarawak. And then we had basically buffer distances at 10, 20, 50, 100, and 500 meters into these different environments. And then again, we dropped the points randomly, and then we went out to sample. There are a bunch of other examples I could give, like in Brazil, for instance, we collected also at certain distances into the forest, but in combination with vegetation greenness values. One of the PhD students I'm currently working with in biology, he's interested in the urban heat island effect on mosquito abundance and diversity. So we used remote sensing to create a normalized built-up difference index, and then use that to drop points randomly throughout the city of Manaus. So there are lots of different ways to do this, but this just shows that fieldwork can really benefit from a geospatial approach. Of course, then there's mapping. Mapping is sort of a no-brainer application of GIS&T in vector-borne diseases. This is just a general map that shows yellow fever vaccination, whether it's recommended or not. So that's at the global scale. Here's a map, and I know this looks a little challenging. This was based on our work in Senegal. And this map shows the vector abundance in females per person per evening of different vectors. So there's Aedes africanus, Aedes dals, and I can't pronounce some of these names. Anyways, Taylori, Vittatus, etc., in the different locations. And again, this is organized by land cover. And so, of course, those of you who make maps know that mapping is always a compromise. And this was a very challenging map to make, because we have these point locations. We're trying to show a lot of things in one place. And so there's quite a bit of symbol overlap, but it does, show some of those patterns. So this is not based on any fancy analysis. This is just based on the abundance data that we collected in the field. So this is a pure and simple mapping application. And then, of course, we can do some, there's a diversity of analyses we can do. And, I won't talk about all of them in part in the interest of time. But, one thing, so this map here is based on some fairly recent data that we collected in Brazil on Zika cases. And we have some monkey trapping sites. And so you can see the monkey trapping sites are here, and they are oftentimes in areas where the Zika case abundance is essentially pretty high. But the points here in this map show just the individual locations where the Zika cases were recorded. And then the saturation here in orange just indicates essentially the kernel density estimate. So we're kind of trying to make a prediction across space. But of course, that's not the only way to display the data. And again, in terms of, well, what's the value of GIS&T in vector-borne disease and landscape epidemiology, well, one of them could just be visualization of data as well. So this map here, and this was just a quick and dirty visualization, was based on the same data, but it was basically as a choropleth map where we aggregated the data by administrative unit within the city of Manaus. Here are the same data represented as a proportional symbol map. So there's usually a lot of experimentation that goes into things, but the point is, we can do kernel density estimates to get a better sense for the distribution, in this case, of Zika cases throughout the city, to identify, where are the hot spots, where are the cold spots. And then, of course, we can do, cluster analysis, look at spatial autocorrelation. So here's an example from the work in Senegal, looking at, just how Moran's I, which is a global spatial autocorrelation metric, how it changes with distance, but then also to see what level of spatial autocorrelation we have at the local scale. So this was based on the local indicators of spatial association. And we had actually set up the sampling design intentionally to try to avoid spatial autocorrelation. And as you can see here, for the most part, we succeeded. All of the gray points indicate that there was no significant local spatial autocorrelation, but there were some areas where we did have some spatial autocorrelation. Anyways, again, with GIS, we can look at those kinds of things. Remote sensing is actually one of my favorite topics. I won't talk about it too much in this talk, but remote sensing is critical because it allows us to generate new environmental data layers that we can use later on for predictive and potentially explanatory purposes. I mentioned earlier that there are a lot of data already available online. I mean, I can go online and download climate data. There are existing land cover data sets that I can download, but they are oftentimes not at the spatial resolution at which I may want the data. And so there's so many different sensors out there right now that allow us to characterize the landscape in a lot of different ways. That would be really useful from a local to landscape type studies. And so here are just a few examples of the products you can generate, like the normalized difference vegetation index, for instance, based on a Landsat image, gives you greenness information at 30 by 30 meters spatial resolution, as opposed to say 250 by 250 meters, or maybe even 1 kilometer. We can generate information about land surface temperature. And again, the student, for instance, who's working in Manaus, looking at the urban heat island effect, I mean, having land surface temperature data can be really quite useful, especially since we can generate them over time and link them to the temporal data of species abundance as well. And then, of course, and this is really critical, land cover. So land cover has a huge impact on species distributions. And interestingly, actually, it hasn't been studied quite as much as one would think, because I think oftentimes a lot of the studies take advantage of existing data layers, and they're limited to climate in particular, happening at coarser scales. But as it turns out, I mean, both the types of land covers that we have in an area, as well as the shapes and the sizes and the connectivity of patches of land cover is really critical to the spread of disease, the persistence of disease. And so this is from our work in Senegal again. So, and it actually kind of highlights both things. It shows the land cover map, but it also shows our sampling points, which were allocated in different land cover types and in these different blocks. Again, we were trying to see how mosquito abundance and diversity changes with land cover across space, and we're trying to avoid spatial autocorrelation. But anyways, one of the things we found was that, distance to forest patches, for instance, was a main driver of mosquito abundance. So having that kind of information is pretty important. Once we have the land cover data, then we can do things like landscape pattern analysis. And again, that kind of relates to this idea of the landscape being made up of patches and being sort of a mosaic, kind of like a quilt, where, I mean, different species have different requirements. Some may require larger patches of forest than others. Some may require certain levels of connectivity, that kind of thing. And so this is an example where morphological spatial pattern analysis was applied to a land cover map, and really the forest portion of that land cover map to see how much of the forest was core habitat versus edge habitat, and where the connections were. And so that information can then later on be integrated in models that try to predict species, try to predict things like risk of spillover. And so that kind of brings us to spatial modeling. There are a lot of different spatial models out there, and I'm happy to answer questions. I'm not familiar with all of them, but I'll mention a few here in a second. But in a nutshell, what most of the spatial models try to do is combine these point level occurrence data of species with the environmental data layers, which are spatially continuous. So they're combined in a modeling framework to ultimately make a predictive map. And typically what we try to do is really, I mean, we want to predict accurately where, let's say, spillover risk is highest, but we also try to kind of get an understanding of the mechanisms behind it. I mean, we want to be able to say that, you know, spillover is most likely to occur if you have a temperature between this and that, or if you're this close to a forest patch, that sort of thing. So we try to understand as well as predict. And those two, unfortunately, are sometimes somewhat mutually exclusive. Not necessarily, but they can be. So here are just a few examples of some of those types of maps that are generated. This is an example from Kramer et al. 2015 that shows a global map of Aedes aegypti distribution, where the red areas indicate the highest probability of occurrence and the blue areas indicate the lowest probability of occurrence. So this was an analysis based on boosted regression trees. They used, Aedes aegypti data from various databases. They got data layers like world climb and other, predictors, and then basically built the boosted regression tree to generate this output. Now, one thing that I want to point out here is note that basically, according to this global map, Aedes aegypti risk in New Mexico, Texas is pretty much zero. And that is clearly not the case. So spatial scale in the modeling process is a huge issue, and I'll get back to this briefly here in a moment. Here's an example from some of the work that we did in New Mexico based on the 2016-17 data. So on the left, you see the CDC map that suggests that Aedes aegypti pretty much occurs continuously throughout the southern 2/3 of New Mexico. Based on the point data that we collected and then various environmental predictors like temperature, precipitation, topography, urbanization, and so forth, this is the map that our student Stephanie Mundus came up with that suggests that there are certain areas where the habitat suitability is much higher than in others. We're actually still kind of working on improving this map as well, but at least it's not quite as generalized as this one. One of the things that we found in the study was that the Aedes aegypti mosquito actually only occurs in urban environments. So we didn't find it in rangeland, we didn't find it in forest land or any of these other land cover types. Now, there are other mosquitoes in these land cover types, like the Culex mosquito, which transmits West Nile virus. But the Aedes aegypti mosquito and Aedes albopictus, which is another sort of peri-urban mosquito, we didn't find them. So we actually did another study in the following years where we sampled in, I don't remember how many cities, but you can see the cities displayed here. And so based on that, oh, and I should say, Stephanie used a maximum entropy model to generate this map. And then I did some work with the urban data using logistic regression to generate these maps. And this was obviously hard to map because these urban areas are pretty limited in spatial extent. So you can just sort of see them plotted here, and then each urban environment is sort of displayed separately. And so you can see that there are certain cities that definitely have a much higher risk of that having that vector than other cities. And then here's another example just to say that, we don't have to be limited just to the present day. It's also possible to use some of the climate predictions to see what might happen down the road. And of course, that's an important topic as well. And so this map from Lippi et al. 2019 shows areas where Aedes aegypti is most likely to expand or contract. So there are a number of challenges with this. One is the ecological meaning of the variables. So when we make these predictions, we may use variables like, say, elevation, but vectors and pathogens, they don't really care what elevation they are. They care more about maybe what the temperature at that elevation might be like. So that's kind of a concern. If we use variables that have pretty limited ecological meaning, that means that we may be able to make good predictions, but we may not be able to understand the process particularly well, which really kind of ties into the next thing. I mean, we can combine tens of predictors and make really good predictions, but ultimately inference is pretty limited. So for inference, what we may want is perhaps more specific direct variables that actually have something to do with, that actually influence the organism. One of the problems with that is that oftentimes the data that we could collect at the local level, like where we collect the species that have that meaning, we don't have environmental layers for them that are spatially explicit across space. So they are not available for prediction. So, sometimes there's a little bit of a conflict between those two, inference and prediction. We kind of have to think about what it is that we are really trying to do. Another thing is that most of the models that have been developed to date are static. In fact, all of the model examples that I gave you were static models. There are dynamic models, and Ben Althaus, for instance, who is on this Create Neo project, he develops those, but they oftentimes lack the spatial component. So, one of the things that we really have to work on is develop these dynamic models that are dynamic across space and through time. Another challenge is spatially implicit or spatially explicit models. So they are models that make predictions across space, but they are not actually considering space as a unique factor. And so the influence of space versus the influence of the actual variables like temperature gets kind of confounded. So ideally, what we really should be developing are spatially explicit models. Another thing that hasn't really gotten much attention is the importance of interacting disturbances. So for instance, and this is not a mosquito example, but let's say if you have what's it called, sudden death, like how does that interact with wildfire? That's two different disturbances sort of happening at the same time. And so not a lot of work has been done on how they interact and how that would influence disease transmission. Human behavior is really something that hasn't been studied much at all either. I mean, all of the work that I've done with my colleagues, I mean, we haven't really incorporated social scientists in the process. We have now clinical studies going on, but to my knowledge, we have never done any work that actually asks people what their perception of risk is, what their behavior is, what they are willing to do. So that is something that I think we need to incorporate much more. And that is certainly something, I mean, in the time of COVID, I mean, wearing masks, wearing them properly, I mean, we've seen how much human behavior influences incidence rates. And so that applies certainly to vector-borne diseases as well. Data are an issue. I mean, like I said, data availability is one thing, but there's now a huge amount of data available. And so we're oftentimes dealing with big data issues. And so, which may require bringing in, and I know the UNM Department of Geography, they have a couple of people that are focusing on big data issues. It's not really my expertise, but that's an area that we have to, address. And then spatial scale, it's always a problem and it continues to be a problem. And just to give a couple of examples here, this is, some of the land cover mapping I've done in Las Cruces with land cover data here showing at spatial resolutions from as fine as 15 meters to as coarse as 1,000 meters. Oftentimes what we see in these models is a mismatch between sort of the scale at which the organism acts and then the scale of the data. And so we have to kind of figure out a way to find the right scale for our models. And so that's sometimes pretty hard to do. The other thing is spatial extent. I mean, the kinds of variables that explain the global distribution of Aedes aegypti or other mosquitoes or other vectors may not explain the spatial distribution or temporal distribution at a much, in a much smaller area. And so in some ways that is, going back to remote sensing. I mean, there are a lot of sensors out there. Some of them have really fine spatial resolution, which is fantastic, but they are limited in spatial extent. Others, cover the whole world, but they have a really coarse spatial resolution. And so dealing with these scale issues, continues to be a huge challenge. And so that kind of brings me to, I think, what some of the future directions are. I think we need multi-scale approaches because some processes at different scales may explain the distribution of an organism at one scale. In fact, there's this idea of hierarchy theory where what we observe at the focal scale is a function of what's happening, not just at that focal scale, but also at the scale above and below. And so, thinking about this in terms of spatial resolution and extent, I think that is something we need to incorporate. So rather than to develop a model, let's say at just a 30-meter spatial resolution, maybe we need to consider variables at different spatial resolutions. I think we really need to take a much closer look at the role of landscape configuration in the spread and persistence of disease. because a lot of landscapes are becoming increasingly fragmented. And that's really a topic that's been largely ignored so far. But like I said, we've done some work in Senegal that suggests it's important, and some other people have done the same. We need to look at joint impacts of emerging disease and other types of disturbances. And to some degree, we're trying to do this in Brazil and Panama, where we have a lot of forest fragmentation going on. Then I think we need to look at the feedbacks between human activities and disease dynamics. Again, oftentimes we model these vectors and spillover risk without really considering the role of people. And I think that's an area that needs to be explored much more. And then finally, I think we need to really continue to promote interdisciplinary research that integrates some of the methods I've talked about with maybe totally new things, like maybe social media. Because ultimately, what we really need to do to effectively manage those vectors is research that addresses both the space and the time component, and that is dynamic, not quite as static as what I've described. And that is all I have.
Thank you for that very, very interesting speech. We'll now open it up for questions from the participants and viewers.
I have a question, Michaela. So it sounds like you have some really interesting projects going on. How has your research changed during the time of COVID? Like, what sorts of things have you had to put on the back burner, or rethink some of your research methods?
Yeah, well, I mean, so for some of my grants, I've requested no cost extensions with the hope that we could, pick up the work this year, which it looks like we will be able to do that. So we've been working on other things in the meantime, but all the field work has been put on hold. The work in this Create Neo project, same thing. One of our collaborators, he's a postdoc. He's now back in Brazil. He's fully vaccinated and he has some personal connections there, and he was really miserable in Texas. So he's back there, but there are still travel restrictions. And so we're kind of hoping that, some of us may be able to do some initial work in the summer, but we're really looking and hoping for a trip in January. So and then a lot of our Brazilian collaborators, they're in Manaus, which of course is a hotspot for COVID. And so a lot of people have died. And so, we're kind of freaked out that we're going to lose some of our key collaborators to COVID, but so far so good. But yeah, mostly it's been the field work that's been put on hold. We've been able to do other things via Zoom and yeah.
Cool, thank you.
Mm-hmm. Would be nice to see some faces. I just see a lot of black boxes with names.
Any other questions?
Oh, I have a question. I was really interested in how you're pulling out urban from natural forest, for your land cover mapping. Have you used things like texture analysis or other things like that to pull out urban from natural environments?
Yeah, I have. And I always try out a lot of different things because you just never really know. So I've played with texture measures. I've played with changing the spatial resolution of various layers and doing a multi-resolution sort of layer stack. I've incorporated spatial autocorrelation measures. So yeah, and sometimes that works, sometimes it doesn't. But yeah. And in some places, like in Brazil, the challenge is to find cloud-free imagery. But once you have it, pulling out urban is actually fairly easy. It's much harder in places like Las Cruces, where a lot of the urban residential areas in particular kind of look like rangelands. So, and then I try a lot of different classifiers usually. So I'm not married to a single one. The other thing that I like to do is use a subpixel unmixing approach instead of trying to just do a pixel-based classification. And I just find that sometimes that works better.
Anyone else with a question?
Maybe put questions in the chat. That's better.
If there are no other questions, I'll turn it over to our Vice President, Brian, for the next part.
Next time I'll ask questions during the middle.
So Brian hasn't joined today. He was on annual leave. So I'll be doing the raffle today. So we have two raffle items. One is a $25 REI gift card, and another is an Esri book. There are a choice of between two books or two titles.
I'll show them.
Yeah, Carol, go ahead.
Here's the first one, Web GIS, although I guess it's backwards, huh? And then the other one is switching to ArcGIS Pro from ArcMap.
All right, so what I'm gonna do is I've took the participant list and I put it in an Excel spreadsheet and gave those numbers. So I put those numbers through a random number generator. And so the first giveaway, the book winner is John Felix. Is John here on the?
Yeah, because we will need his mailing address.
Right.
It looks like he's on, but maybe he's not.
So yeah, the name is still there. Hey, John, if you could hear me, please send your email. And our next, the $25 REI gift card the winner is Sujan.
Yay.
Thank you.
All right, so I have your email, so I will get that. So that's all, and thank you, Michaela. This is great, very interesting presentation and research. Thank you for doing the talk.
Yeah, thank you all for coming.
Yeah, thank you, Michaela.
And so before we sign off, any other last questions or anything? All right, I don't see anything, but hey, John, I don't have your contact, so if you could send us your information, we can get that book mailed to you.
Hey, Sandeep, it's Sigurd. I work with John, and I can make sure that the connection gets made.
All right, thank you, Sigurd.
Either that, or I'll take the book for him, or the gift card.
That works too. All right, everyone, have a good rest of the day, and see you next Thursday for our third talk on COVID dashboard by real-time solutions. All right.
Thank you.
Bye all.
Bye.